Okay, hi everyone. Let me just make sure I change my settings so I can see your questions. And we'll go ahead and get started. This is our Thursday version of the, hello again. This is our Thursday version, so ask away if you have questions for tomorrow's quiz. Let me see, what can I... Well, in case you didn't watch last night, just a reminder again that the quiz will cover only pages 132 to 149. Sorry, looks like my glasses are creating quite a glare there. Maybe that's better. Okay, so yes, can I explain how to graph hemoglobin and why the fetal acid endohemoglobin? So the idea is that no matter what, hemoglobin is always going to give oxygen away when there's not very much oxygen around, like when it, and then it's going to take in oxygen when there's a lot there, right? So think of it as very practical. Hemoglobin is not a selfish molecule. It's not going to hold on to its oxygen if other cells around it need it. That being said, so no matter what, we have that, oxygen binding curve, right? Where we can see that depending then, if on the x-axis you can see the partial pressure of oxygen, then depending on the partial pressure of oxygen in that area, that will determine then how saturated, meaning how many oxygens are around hemoglobin, right? So what we see, generally speaking, is that when there are, there's not a lot of oxygen around, there's a low partial pressure of oxygen, the percent saturation is low. But as you move over and the partial pressure of oxygen is higher, meaning there's a higher concentration essentially of oxygen, then the hemoglobin becomes more saturated. Okay, so that's our general curve no matter what. The thing is, there are various conditions that will change how grabby hemoglobin is for the oxygen, or what we call the affinity for oxygen. So in the case of, well, I'll just start with, so you have your standard adult hemoglobin, right? But that also assumes, that it is at a certain pH, right? When the pH goes down, then that's called the Bohr effect. And that's essentially going to change the affinity or the grabbiness of hemoglobin for oxygen, right? When the pH actually goes down and it becomes more acidic, then we see the entire graph shift. And what we see then is that hemoglobin will now not bind to oxygen as well, meaning it'll let it go more easily, right? So let me see, let me pull up my graph to make sure, and if you have it, you can certainly look at it in your notes too. But essentially you can see that the graph 7.4 is the dotted line and 7.2 is the solid line. Excuse me. And you can see that essentially that at this, now at the same partial pressure of oxygen, so let's just pick like 40 and we trace up, right? I can see that the hemoglobin, excuse me, at 7.2, pH 7.2, is less saturated, meaning carrying less oxygen than at pH 7.4. So what that's telling me is that the whole graph is shifted to the right, right? That at the partial pressure of oxygen, but a lower pH, hemoglobin doesn't hang on to oxygen as well. Again, you could just memorize that, but think about what that means, right? It means that the hemoglobin is letting go of the oxygen more easily at that point in time. And it actually makes sense why, because when you have not a whole lot of oxygen, you're going to have, now that means more CO2. More CO2 would mean more carbonic acid, which is a intermediate in the process of converting the carbon dioxide into bicarbonate. And so as a result, you would end up lowering the overall pH. So it makes sense that a lower pH would signal to the hemoglobin that it needs to let more oxygen out because it's what it needs. For the fetus, so that's acid versus like standard, right? For the fetus, it's the idea that now, so anytime you have a shift to the left, it means it grabs the oxygen better. If you have a shift to the right, it grabs the oxygen worse or lets go of it more, right? So in that case, what you end up with is for a fetus, the only source of oxygen for a fetus is its mother. So it has to essentially be able to grab its oxygen from its mother. And that's why the fetal hemoglobin actually has to have a higher affinity than the adult hemoglobin. So you're all born with, or you have fetal hemoglobin before you're born. And then slowly, slightly after you're born, you convert it to adult hemoglobin. Okay, let's see. Let me go back. I know I missed a bunch of questions while I was talking about that. So why do reptiles want to bypass the lungs with their secondary organ when underwater? Okay, if it bypasses them. Okay, so yeah, essentially, um, it's the idea that yes, Schweitz is exactly right. Ideally, you want to go to your lungs and get oxygen, right? But for an animal who's swimming underwater, it's going to make more sense to say, well, I'm not going to get any oxygen right now, even if I take it to the lungs. So I'm going to bypass lungs, go through my second order, send the bad blood back to the body, and in doing so, still be able to get some oxygen. Because the thing you want to remember is when we talk about oxygenated, like deoxygenated, oxygenated blood, it's not like you deplete all of the oxygen in one round of going to the body. In fact, most of the time, like deoxygenated blood could still be like 75% oxygen, right? So to send it back to the body now just means you can now get rid of some more of that oxygen and give it away to the cells, as opposed to sending it up to the lungs and not really picking up any oxygen anyway. Um, 1H on the practice quiz. Yes, let me pull up the practice quiz for a second. Um, and at the same time, I guess I'll talk about mixed blood with amphibians. So, mixed blood actually isn't an advantage, right? I mean, it's what it has and it's fine, it functions. But if we're comparing separated blood versus mixed blood, then separated blood is going to be preferred because, again, it's the idea that now you're not sending partially oxygenated blood back out to the body when it hasn't had a chance to go back to the lungs and get some oxygen. So, is it functional to have mixed blood? Certainly it's okay. But it's actually probably more advantageous to have separated blood because that means you've sent it to the lungs and got oxygen, right? And it's not going to have any situation where it's going to mix with blood that is deoxygenated, which is what you would have in the shared ventricle or even the partially shared ventricle that you see in the reptiles. Okay. Let me pull up that practice quiz though for a second so that I can answer Sonia's question. Okay, let me see here. Let me just find a window that I can open it through though. Okay, sorry, this will take just a second. Let's see, maybe while I'm waiting, since it's still a bit, okay. So 1H, it says, suppose the level of partial pressure of oxygen in lungs is measured 70. The atmosphere outside the body must be. So in this case, is what you want to kind of think about is how might it compare, right? So there's other pieces to it, obviously. You're going to see, there's water vapor actually that contributes to the partial pressure, and that actually reduces the partial pressure a little bit. So you'd expect that in this case, sorry, let me pull this up for a second. So you would expect then, because the water vapor in your lungs now sort of adds to it, that actually outside the body, you would see a partial pressure of oxygen greater than 70 millimole, which is because that water vapor kind of changes the composition of things. But I wouldn't really worry too much about that. I didn't stress that in lecture. So this year, I wouldn't really worry about that. In other years, I may have stressed it more. Okay, where we go? Let's find your questions again. Okay, so we talked about mixed blood then. Okay, counter current exchange. Yes, let's talk about that. So with counter current exchange, it's again the idea that regardless of what direction the fish, I'll just use that particular example since that's what we talked about in class, is swimming, you have the blood in the gills going in one direction, and then the oxygen or the water is flowing across the gills in the opposite direction. Really, what that's going to allow you to do then is to have a situation where, um, simply put, your blood, or not your blood, the fish's blood is always coming into contact with more oxygenated water than it previously did, right? And so, because of that, the whole length of the gills, you're able to diffuse oxygen from the water into the gills. Whereas if you look at the picture in the PowerPoint of concurrent flow, where they're going in the same direction, pretty much after like half of the gill, then at that point they both are at 50% saturation or 50% oxygen. So that means that you're going to have essentially no real gain of oxygen across the second half of your gills, so that really makes them not very useful anymore. So that's sort of countercurrent flow is just that general idea. Let's see. Okay. Why does smoking raise LDL and lower HDLs? To be honest, I don't know that they have the full answer to that. We definitely see that it occurs. Let me pull this up here so I can make sure I have the right slide that you're asking about. Sorry, let me pull that up here. So essentially, let me see if I have any extra notes in here that I can. So simply put, at least with smoking, what they found out is that there's a compound in cigarette smoke that's called acrolein, and so when the body's exposed to acrolein, it actually seems to interfere with HDL. So that's probably how it sort of messes up HDL levels. They say acrolein also like interferes with LDL's ability to basically keep together, right? And so I think essentially probably the just the interaction of those compounds with processes in the body probably make those levels go. But I don't know that they have an exact knowledge of why either of those things happen. It's just a good example of how it's not just all genetic, right? That there certainly is a lifestyle component to that, and that's all I would really want you to know. What's the difference between lymph and interstitial fluid? So interstitial fluid is technically the fluid that's like outside your cells and sort of the thing that is exchanging with the fluid inside of your cells, right? Your cytoplasm, etc. Lymph is specifically the fluid that's flowing through your lymphatic system. So they're probably very similar in composition, to be honest. It's a little more the idea of like where you're finding them. And so it's probably not exactly the same, right? But it's similar in that they're sort of both fluids that are not blood that at the same time are participating in that exchange, all those exchanges that are happening. Okay, what is the Bohr effect? The Bohr effect is the idea that at a lower pH, hemoglobin will not bind to oxygen quite as well. And so because of that, the affinity for oxygen goes down. I talked about it a little earlier in the lecture as well. So it's probably best in time-wise if we kind of don't necessarily repeat it right now, but I think it's it's one of the first or second things I talked about. Micelles. Micelles are essentially what you get when you take a big blob of fat and break it into smaller droplets. So the micelles are what the micelles are essentially what the bile will break the fat into. It's not a term I'm super concerned that you know, though, to be honest. So, certainly it's not a bad thing to know, but I'm not gonna, stress it in any way, shape, or form. So micelles are essentially what you get when you break them down. Technically, I guess after the lipase works, but like I said, I'm not super concerned about that term. Do you need to know what the component of blood is and what they're made of? Yes, you should know. Well, I mean, I want you to just know the general components of blood, right? That you have your red blood cells, your white blood cells, your platelets, and your plasma, right? The idea that red blood cells carry oxygen, white blood cells are for immune system function. The plasma contains mostly water, but also dissolved proteins, and in this case, bicarbonate, and all sorts of other things like that. Hormones, etc. And then also that finally you have the platelets, and the platelets are involved in the clotting reactions that help essentially seal any holes that might not erupt, but form due to damage or something like that in the blood vessels. Okay, let's see. Have I gotten any other questions? No? Okay. Let's see. Other questions? I don't think I've seen any notes pop up. Well, let me take a look at the practice quiz, I guess. Let's see if I notice anything. I guess. Oh, I guess something to mention is the last question, the herbivores, and the cows and the stomachs. That was actually on the case studies that I gave you. It's not something I stressed in class though, so don't worry about that. We used to teach it in class and then we took it out. So, oh, okay. So let's see. 9C on quiz 2. Okay, hold on. Let me look up quiz 2 then. Give me a second. It's pulling up here. Okay, so 9C. Let's see. Okay, so 9C has to do with deep vein thrombosis. Let me read the question real fast, make sure. So it's really just asking about how blood is moving through your veins, right? Because remember, the big issue with your veins is that you don't have any real pumping, like pushing force from your heart. It's so far away from the actual pump of the heart that you need something else to help push the blood up, right? And so you would essentially need your skeletal muscles around to be moving to push that blood up. And if you're sitting still, like in an airplane for a really long time, that's really not going to work, right? So in this case, I would want you to check all that apply, right? So the two in this case that would apply to veins would be the first one. The skeletal muscles around the veins push blood back towards the heart, because that is true, and that's why you want to walk around um, when you're on a flight, or have those compression stockings that help essentially have those muscles squeeze on the veins and push the blood up. And the third one, which is valves in the veins prevent backflow of blood, right? Because those two things combined are really going to help push the blood up to the heart when the veins don't really have a whole lot of, I guess, help from their own, you know, situation. Okay, let's see. The mammalian diving reflex, also something I don't need you to know on the quiz, but really quickly, it is a good example of that really close interplay, though, between the respiratory and circulatory system, because it's the idea that um, when you have an organism, a marine mammal that's diving and they're going to be underwater for a long time, they really want to kind of maximize their oxygen. And so as a result, they don't want to be sending blood and oxygen to their hearts or to their body cells super fast, and then like depleting their oxygen too quickly in case they need it. So um, instead, what they do is they slow their heart rate down, like super, super slow, right? And in doing that, and they have other, strategies as well, like they have a lot of myoglobin, they shut off blood flow to other areas, but it's the idea that in doing all those things, they're going to slow down the depletion of oxygen so that they can make it last for longer should they need it. 26 on the practice quiz. Let me see. Yes, I can. Let me find it. Okay, since the second one doesn't have a 26, I will assume you mean the first one. So yeah, carbon dioxide that's produced from the tissue, it's sort of just asking you, since I know the carbon dioxide, which is the waste products of cellular respiration, need to leave the body and go to the lungs somehow. I really want it to, you want to know how it's carried, right? And so whereas oxygen is carried in the hemoglobin, the carbon dioxide actually has to get converted by the red blood cells into bicarbonate, but then it's dissolved in the plasmid as bicarbonate. So the correct answer there would be C. And the enzyme that does that converting is called carbonic anhydrase. Okay, can I go over the deoxygenated oxygen blood flow in the heart? Which blood would go into the atrium? Okay, yeah. So it's a loop. You can start wherever you want. But in this case, I guess I would start where where the blood comes to the heart from the body. So it would be deoxygenated when it enters the right atrium, right? And it would be coming into the right atrium from the superior and inferior vena cava. That is deoxygenated blood, right? It's delivered its oxygen to the body cells. Now it has to come and get more. From the right atrium, it would go through the tricuspid valve to the right ventricle. And then the right ventricle would pump it out to the lungs through the pulmonary artery to get oxygen. So at that point, it's all deoxygenated. Another way of thinking about it is everything on the right side of the heart is going to be deoxygenated. Then it would go to, and so that includes the pulmonary artery, right, to have deoxygenated blood. Then it would go to the lungs, pick up oxygen by diffusion, when it goes through those, when the blood vessels go right next to the alveoli, right, and then it would come back to the heart through the pulmonary vein. The pulmonary vein then would carry oxygenated blood, and now it would dump into the left atrium. From there, it would go through the bicuspid valve, which is also called the mitral valve, and end up in the left ventricle. So again, you can also think that on the left side of heart is where you have the oxygenated blood, right? From there, then the left ventricle would pump, send the blood out to the body cells by going through the aorta. So hopefully that helps. Right side, deoxygenated or oxygen poor. Left side, oxygen rich. Okay, can I explain the circulatory system for animals who have mixed blood? Yeah, so the two animals where I mean, in our context, right? There are others, but in our context, reptiles and amphibians are the ones who would technically have mixed blood. And what that means is there is not a distinct separation between oxygenated blood and deoxygenated blood, right? Let me start by having you think about like our hearts, right? In your heart, the deoxygenated or oxygen poor blood is kept completely separate from the oxygenated blood on the right? Because the wall of our hearts goes all the way up. So there is complete separation of oxygen and deoxygenated blood. Whereas I'll just start with the amphibian, right? They have two atria but only one shared ventricle. Think about their flow of blood. The deoxygenated blood goes into their atrium, then it goes up to their lungs, picks up oxygen, dumps into the ventricle, but there's only one ventricle, right? So now it's going to pump from that ventricle back out to the body. Um, I'm sorry, let me go back. It starts in the um, right atrium, right? Then goes to the ventricle, then it goes up to the lungs and gets oxygen, comes back to the left atrium, but then goes into the same ventricle. So you have a mixing of oxygenated and deoxygenated blood. So that's okay, but not ideal, right? Because you might be sending blood out to the body that's not necessarily oxygenated, and then you could be sending blood to the lungs that's already oxygenated, doesn't really need to go there. The reptile gets a little bit better with that, where you have a partially separated ventricle. So now it's like some mixing, but not as much, right? But then ultimately, ideally, to have separated blood is going to be a more efficient way of moving the blood around. Okay. Other questions? Let me see. No problem. Let me take a look too to see if I see anything else that I should mention. There is a question on one of the practice quizzes. Sorry. That has to do with, I think, the excretory system. And just so you know, like, you don't need to know that. I think it's 16 um, because it's not on this quiz, so don't worry about that. Let's see. What else? Think I see anything else here. Remember that you do not need to know again the stuff before 132, right? You will see that show up on your final exam, but not for this quiz. Yes. Okay. So blood pressure essentially is a measure of how hard your heart is working, right? I mean, the idea being that your heart is pumping, and if it's like pumping really hard, then it's going to push on your blood more, right? So blood pressure really is a measure of how much force are you exerting on the walls of your blood vessels, and that becomes concerning because um, too much blood, like too much pressure from your blood, is going to make it such that the inner lining of your blood vessels is getting kind of pummeled by that pressure, right? Think of like spraying something with a garden hose. So if you have now high blood pressure, that could damage the inner lining of the blood vessels, which would then lead to atherosclerosis. That being said, when we look at blood pressure, there are two numbers. The higher number is what we call our systolic pressure, the lower number, which is our diastolic pressure. And it's the overall idea that when your heart's pumping, it's going to exert a little more pressure, right? Think of like when you squeeze on something, you're exerting more pressure. So that number is what we call our systolic pressure, because it's saying when your heart is physically contracting and pushing the blood, how much pressure is now the blood going to apply on the walls of your blood vessels? But your heart is not constantly contracted, right? It contracts and relaxes and contracts and relaxes. So, that being said, in between contractions, when it is relaxed, there's still going to be sort of a natural baseline amount of pressure that's on your blood vessel walls, and that's what we call our diastolic pressure. Blood pressure can be controlled by a lot of different things, right? I mean, certainly hormones and whatnot, right? But it can also be things like the arteries themselves. And you measure blood pressure in an artery, right? So if you're measuring how much pressure is on the walls of those blood vessels, there's a lot of factors that contribute to that, right? It could be, for example, when you dilate your blood vessels, like you make them wider, then the amount of force on the walls of the of the blood vessel is less. Or if you make it narrower, right? Like if it's really cold and blood vessels constrict, that might increase the pressure because now it's a smaller space, but the same amount of blood is trying to get forced through there. It's a higher pressure, right? Remember, they have to be able to kind of like stretch with your blood vessels, right? So if they're more rigid, that might also make it such the pressure is increased. Um, I don't think, I mean, I think that likely it's effectively no. I think veins have their own issues, but atherosclerosis is typically due to damage to the blood vessel walls. And so, given the low pressure in the veins, at least that seems like it would be highly unlikely. Technically, could it happen? I guess so, right? But I don't think it really does, typically, at least. Because atherosclerosis really has to, it's the plaque deposits building up there. So, again, I suppose it could be, right? But those plaque deposits tend to build up because you've damaged the lining of the blood vessel walls, and now it has this whole process where like plaque builds up underneath of it. So, you want to know atherosclerosis more in the context of for arteries and the fact that if you have atherosclerosis, now you have a situation where it might um, totally plug up, right? Because atherosclerosis doesn't mean the blood vessel is plugged, but the risk now is that that plaque has a really kind of thin membrane on it. And so when you um, basically rupture that plaque, it triggers a clotting reaction. And a clotting reaction is going to now potentially plug up a fairly small blood vessel because the clot will block all the flow. And that's when you get something like a heart attack or a stroke. Okay, any other questions? Let me see if I can take a look and see what else I see here. You'll notice that, in question 27, I do use the terms for the animals that are a little more technical. So, just be familiar with those, I'd say, also for the APs. Yeah. Um, do we need, so I would say yes, I want you to know enough of how the clotting process happens, like not like every itsy bitsy detail, but know that sort of whole cascade, right? So the idea that you have damage to the blood vessel walls, let's say you've cut yourself, right, and that exposes collagen fibers, that makes the platelets more sticky, and those platelets now stick to that area and they'll start to release clotting factors. The clotting factors will now make prothrombin, which is already in the blood, convert into thrombin. Thrombin will now take fibrinogen, turn into fibrin, form those sort of fibers of fibrin such that now it's going to help better patch up and trap things to plug up a hole in the blood vessels. Heartbeat ultimately is controlled it originates in the heart itself, right? But it can be controlled then by various things like hormones or, um, other factors, nervous system, and things like that. So I guess it's like two levels of control, right? There's the simple act of making your heart beat, which would be internally controlled by the heart itself with the SA node firing and sending a message to get the heart to beat. But then there are other factors like, like I said, hormones, etc., that would make it go faster or slower. Remember that the heartbeat is an electrical signal that's passing through, right? Remember that you essentially have the top and the bottom of the heart. So when your heart contracts, it's like the top of it squeezes and the bottom squeezes, right? So the top of the heart contracting pushes all the blood from the atria into the ventricles, and then the bottom portion, the ventricles contracting together, makes the heart go out to the lungs on one side, to the body cells on the other side. Let me see and make sure I didn't miss any questions here. Really, the role of the bundle of His and Purkinje fibers is it's like part of the like chain reaction, right? That's all I really need you to know about that. Then that the SA node sends a message to the AV node sends a message to the bundle of His, which sends a signal to the Purkinje fibers, which now makes the ventricles contract. No, I mean, I would say for the AP, I would know the list of animals that I want you to know. It is in your notes, actually. Let me see if I can find the page for you for a moment. I might have the file already pulled up. So just to help you like remember the names, it would be on page 129. There's actually this like diagram from your book where it says like selected animal phyla, and it just tells you like the names of the phylum and some of their the technical names as well, what they are. Okay. Oh, no problem. Other questions? Let's see. What other recommendations can I give you guys? I think yesterday night there was a question about blood sugar and whether or not you need to know that. So just to clarify, yes, you do need to know that. You need to understand sort of how insulin and glucagon work. You need to also be able to tell me how the other hormones like gastrin and CCK and whatnot work as well. And remember, we are not formally talking about the endocrine system like individually all by itself. So instead, you kind of just see it get peppered in through different parts of the circulatory, respiratory system, and digestive system. How does CCK work? So essentially, what CCK is, it's a hormone, right? So when you have CCK, what it actually does is it will be released by the small intestine cells as a result of food coming into the small intestine. And then remember, a hormone's made in one place and used in another. So in this case, now the CCK is going to go and act on the gallbladder and tell it, hey, I need some bile. It's going to go to the pancreas and say, hey, I need some digestive enzymes. So in now going to those organs and binding to those cells, it's going to trigger those organs to release bile and release digestive enzymes into the small intestine so that you can finish off digestion. You're welcome. Let me see what else. Let's see. Oh, okay, gastrin. Gastrin's another hormone. Remember, for that particular diagram, which you have, let me check your notes so I can tell you which page that is. So gastrin, which you're going to find on page 137. Remember, everything in that diagram where it says control of human digestive system, everything that's all capital letters is a hormone. So what happens is there are certain cells in your stomach that will release gastrin when food comes into your stomach. And it's basically now your gastrin will go and tell the other cells of your stomach, like, hey, I need more HCl, hey, I need more pepsin, increase the movement of the stomach. So it kind of just helps moving things forward to make digestion happen. Let's see. What was I talking about before? I will see if there's. Oh, no problem. Let's see. Sorry. Let's see. What else can I advise you about in the last 5 minutes here? Any other questions people have? Make sure you understand, like, structurally how the different blood vessels work. What about them allows them to do their jobs, right? So the whole idea of structure and function. Know about the pathways that things take. Remember, there are a lot of tools on the website for you. Obviously some of it you won't need yet, but let's see what else. Let's see, what can I tell you guys about? Let's see. I talked about oxygen binding curves. Definitely know the systems. I think I already mentioned it before, but understand how each of the systems, digestion, circulatory, respiration, each, not just in humans, but in other organisms, right? Let's see, you have about 2 minutes left. So again, just make sure you ask any questions you might still have. Any last minute questions? Can't really think of anything else to tell you all right now. Definitely be able to read an oxygen binding curve. I think I already mentioned that in units in class. No, your quiz information starts on page 132. So beginning of digestion, not 129. 129. Yeah, 129 is 129 is like all the general animal stuff, which again you'll see on your final, but not for this quiz. Oops. Okay, so it looks like people are done with questions. I mean, I don't know, I don't see any more popping up. And our time's nearly up, so I guess last call if you guys have any questions. Otherwise, we'll probably just call it a night soon. Yeah. Okay. So I guess. Oh, you're welcome. Just let me know if you guys have questions. I'll be up for a little while still, um, working on some stuff. So if you have questions, you can email me. You can come tomorrow during tutorial and ask questions. And we'll just, let's answer them as we come then. Okay. Um, and then, yeah, have a good night and get some rest. And eat a good breakfast in the morning. Otherwise, like I said, just email me if you have questions and I'll be happy to answer them. All right, guys. Have a good night. See you all tomorrow.